Hello dear students. Today I am going to demonstrate you the experiment number 4 of the subject pharmaceutical microbiology. The object of the experiment is to perform different staining methods including simple staining, gram staining and negative staining. So today I will be demonstrating you how we can perform the simple staining experiment and gram staining experiment in our laboratory. We know that the staining procedure plays a very important role in the visualization of the microorganism, especially your bacteria, because the bacteria is having somewhat transparent or semi-transparent structure. So that's why under your light microscopy or the compound microscope, these microorganisms are not clearly visible. So, if we impart a specific color to these microorganisms, then the features of the bacteria can easily be visible. That can help us in the characterization, identification of different categories of the bacteria. We also know that these staining methods have different categories, like the simple staining method is available, gram staining method is available, differential staining method is available, or the acid fast staining method is also available. So different methods of staining is available. So as per our curriculum, we need to perform the simple staining and gram staining method. So one by one, I will demonstrate you that how we can perform the simple staining procedure and gram staining procedure in our laboratory. And while performing the experiment, I will also explain you what is the exact principle that lie behind that particular staining method. So the first staining method that I am going to demonstrate you is a simple staining method. Now, so for performing the simple staining method, we need certain things. The first thing is actually the slide over which will perform the staining method. We also know that the simple staining method can be defined. It is the method in which only a single category of a dye is used. It means only one dye we have to use in case of the simple staining. That dye can be any basic dye, including your methylene blue, crystal violet, carbon saffronin. Any basic dye can be used for this purpose. So today I am using this methylene blue dye for performing this simple staining method. So first of all, we have to take a very neat and clean slide. Suppose if you are performing this experiment in your laboratory, the first step is you should clean the slide, okay, because if some lipid type of things is available, some greasy material is available over the slide, then that things will not make this simple staining procedure very convenient and your microorganism may not be visible clearly under your microscope. So the first thing is your slide should be very clean, okay. Now the next step is you have to take a loop full of microorganism or the bacterial sample that we need to visualize under microscope, okay. So then we have to make a very thin film of this sample of microorganism on the surface of the slide, okay. This is the first step, that first you need to take the slide. The slide should be very clean. Then what we have to do, we have to take a loop full of culture or the microorganism sample. We know that this device is actually called as inoculation loop. So with the help of the loop that is available on the inoculation loop, we have to take a loop full of the sample and we have to spread that sample on the surface of the clean slide in the form of a thin layer, okay. This step is actually called as the formation of the smear. We have already seen in case of the theory also that in many experiments we have to first go for the preparation of the smear, okay. So a thin sample is applied of the microorganism on the surface of the slide in order to form a smear, okay. Then we have to go for the air drying of the smear. Then after the air drying mechanism, what we have to do, we have to go for the heat fixation of these microorganisms on the surface of the slide. So for the heat fixation, what we have to do, we have to repeatedly pass this slide over the surface of the flame like this way, okay. Only for maybe for 5 to 6 times, or maybe for few seconds we have to pass our slide over the surface of this flame, so that because of the heat, the structure, because that exposure is very brief, so that the structure of the bacteria may not be deteriorated because of the application of the heat, but also the microorganism or the bacteria will be fixed on the surface of the slide. So once the heat fixation is over, then what we have to do, we have to go for the application of the selected stain. Like today I am using methylene blue on the surface of the prepared smear. This means we have to go for the application of the selected stain on the surface of the smear. Okay, then we have to wait for some time, okay, because the stain needs some time so that can easily color the microbial sample. Okay, so we know that in case of the simple staining we are using different categories of the dye. So every dye is having a particular exposure time. Maybe for example, if you are using methylene blue, then we have to at least wait for 1 minute. Okay, then we know that in case of simple staining, the dyes that are used are generally the basic dyes. Okay, so these basic dyes reacts with the acidic component of the bacteria in order to stain it. Okay, so after a particular period of time, after waiting for 1 minute, what we have to do, we have to wash the stain with the help of the distilled water. Okay, so when we are going for the washing of the slide, then the thing that we need to remember is when we are applying the water, then the stream of water should be parallel. It should not be perpendicular, because what happens, maybe you can wash this with the help of the tap water also, okay, but still if you are using the tap water, then the stream of the water should be parallel, because if you are using a perpendicular stream of the water, then the microorganism which are already fixed on the surface of the slide may be oozed out of this slide. Okay, then nothing will be visible under your microscope. Okay, so we have to use the parallel stream of water in order to wash the slide. Okay, then what we can do, we can go for the drying of the slide. Maybe sometime we can go for the use of some tissue paper or some filter paper in order to dry the slide. Otherwise, with the help of the air drying mechanism also, we can simply dry the slide, and then we can visualize under your microscope. And as you know that in case of the simple staining, it will not differentiate different classes of the bacteria, but whatever the microorganism, whatever the bacteria that are available in the sample will be clearly visible under your microscopy, and you can easily identify or differentiate between like the morphological characteristic of the bacteria, like the sample of the bacteria consists of the coccus, or it is having some bacillus structure, or maybe it is having certain spiral bodies. Okay. Now coming to the next staining procedure, which is called as the Grams staining. So I am also going to demonstrate that how we can perform the Gram staining in our laboratory. We have already discussed this in our theory portion, that this Gram staining is actually the differential staining procedure in which we can differentiate the microorganism in two categories as Gram positive bacteria and Gram negative bacteria. Okay, so let us see how we can perform the Gram staining experiment in our laboratory. Okay, so the first step is same as in case of the simple staining, that is slide that we are using for this purpose should be very clean in nature. Okay, so first we need to take a very clean slide. Okay, then next step what we have to do, again we have to take a loop full of culture. Okay, and we need to go for the preparation of a very thin layer of this culture in order to produce a smear. Okay, then what we have to do, again we have to wait for some time so that the smear will get air dry. Okay, now we know that in case of the Gram staining method, different number of dyes are actually used. Okay, these dyes are the primary stain. First we have to go for the use of the primary stain, then we have to go for the use of the moderate. Okay, then we have to go for the use of the decolorizing agent, and at last we have to go for the addition of the counter stain. As in case of this simple staining, we are already using only a single dye or a single stain, whereas if we are going for the use of the Gram staining procedure, in that case multiple dyes are used. Okay, So now this smear is air dry. Then what we have to do? We have to go for again fixing of the smear by passing it over the flame for maybe for 4 to 5 times or maybe for some fraction of second, so that whatever the bacteria that are available in our sample will get fixed on the surface of the slide. Okay. Now your sample is ready over the surface of the slide. Now we can go for the performance of the staining procedure. Okay. So the first stain that we need to use in case of the ground staining is crystal violet. Okay. So first we'll take the crystal violet dye and we'll put it on the surface of the smear. Okay. Like this. Okay. And again we have to wait for some time. Approximately we have to wait for 1 minute. Okay. Now here the crystal violet will act as a primary stain. Okay. So whatever the microorganism that are available in our sample will be stained violet because of the presence of the dye, that is the crystal violet. Okay. We can also suppose if we are performing a experiment of simple staining, in that case also we can simply go for the use of the crystal violet as a single dye, again wash it and then it can be visualized under your microscope. Okay. So crystal violet, if singly it is used, then we can call it as a simple staining. But after crystal violet, if we are adding gram iodine, if we are using saffronin, it means we are actually going for the ground staining procedure. Okay. Now after the addition of the crystal violet and waiting for some time, then what we have to do? We have to go for the addition of the second stain, that is actually called as the gram iodine. Okay, we have to go for the addition of the grams iodine. Okay, because we know that we have already covered in case of our theory that when we go for the addition of the crystal violet and then grams iodine, that results in the formation of a complex which is called as CVI complex. Okay, so whatever the cells that are available, maybe it is gram positive or maybe it is gram negative, then both the categories of the cell will result in the formation of the CVI complex. Okay, then here the CVI complex is actually preparing and again we have to wait for some time, because we know that in case of the gram staining we are differentiate the bacteria into two classes, one that is the gram positive bacteria and one which is a gram negative bacteria. But once we go for the addition of the crystal violet and then we go for the addition of the grams iodine, all the categories of the bacteria, either it is a gram positive bacteria or a gram negative bacteria, both the categories of the bacteria will be stained and will result in the formation of the CVI complex. Okay. Now, once the CVI complex will be formed over the surface of the slide, the next step is what? We need to go for the washing, okay? Or maybe directly we can go for the decolorization of the slide, okay? With the help of some decolorizing agent, okay? Maybe we can go for the use of 90% ethyl alcohol, okay? So we have to go for the decolorization of the slide, okay? So like this way, we'll go for the decolorization. Again, we have to keep in our mind that while we are decolorizing, we need to remember that the flow of the decolorizing agent, it should not be perpendicular, it should be parallel, okay? And the decolorization procedure is actually continued till you will find that whatever the crystal violet color that is available on the surface of the slide, that should be somewhat, it should be diminished, or it should be something like it is not very clearly visible on the slide. Okay, like this way, as you can see here, that initially the color was very bright, okay, crystal violet color was very dark, but now because of the decolorization, it is somewhat diminished. Okay, so what will happen in that case? The gram negative bacteria, okay, they will loses the CVI complex, because we have already discussed there is a morphological difference, there is a difference in the structure of the gram positive bacteria and the gram negative bacteria. Okay, so because of a very presence of a very thin cell wall in the gram negative bacteria and because of the availability of the high lipid component in the gram negative bacteria, the lipid component, because of the dissolution of the lipid component of by the help of an alcohol, the CVI complex can easily lose by the gram negative bacteria. Okay, whereas if we talk about the gram positive bacteria, then they will retain their CVI complex, and because of the availability of the CVI complex, the gram positive cell will appear something very violet or something very near to black color. Okay, but now because of the application of the decolorizing agent, whatever the gram negative cells are available, they are decolorized. Okay, now next step what we have to do, we have to go for the addition of the counter stain. Okay, so here I am adding the counter stain, and what is the counter stain in case of the gram staining? It is safranin. Okay, then we have to go for the addition of the safranin, and again we have to wait for some time. Okay, so why we are adding safranin here? Because we know that the gram negative cells are now become colorless. Okay, so if you want to differentiate the gram positive cell with that of the gram negative cell, so these safranin dye will stain the decolorized gram negative cells. Okay, so the gram positive cells, they are already stained because of the presence of the CBI complex. They are already visualizing in the form of some violet or blackish violet color, but when we go for the application of the counter stain or after the application of the decolorizing agent, the gram negative cells are now colorless. Okay, so if we want to visualize or we want to differentiate the gram positive cell with that of the gram negative cell, we need to counter stain these slide with the help of some counter stain like safranin. Okay, then after the application of the safranin, we have to wait for some time, and again we need to wash the slide with the help of the distilled water. Okay, carefully we need to wash. Okay, and then again we have to dry this slide and then visualize under your microscope. Okay, so here in this slide you will find that two different categories of the bacteria will be visible. One that is having somewhat violet or blackish violet color, they are the gram positive bacteria, and second will also visualize the presence of some gram negative bacteria, and because of the dye that is safranin, these gram negative bacteria will be visible in the form of some pinkish red color. So in the same slide you will observe two different colors of the bacteria, one that is violet or blackish violet color and one that is having the pinkish color. So the black bacteria or the violet bacteria, they are the gram positive bacteria, and the pink bacteria or reddish pink bacteria, these are actually the gram negative bacteria. Okay, whereas if we talk about the simple staining, then for example if you are using methylene blue or crystal violet, so whatever the color of the dye is available, all the microorganism irrespective of its class will be visible in the same color. Like in case of methylene blue, all the bacteria will be observed or will be visualized as a blue color bacteria, and definitely with the help of the simple staining also we can go for the characterization and morphological differentiation of different classes of the bacteria. Thank you so much.